The purpose of this video is to understand what polypeptides are, how they are related to our understanding about amino acids, and what are peptide bonds. So in 12.3, I want to begin the video by giving an analogy of a beaded necklace. So when you look at this beaded necklace, the beads could be compared to amino acids, and when these amino acids are linked together in a linear chain, we call the entire chain as a polypeptide. A single polypeptide is made up of more than 10 amino acids linked together in a linear fashion. Now, if we look at the links, we call each link or the bond between each amino acid as the peptide bond. So, this is a simple way of understanding how polypeptides are formed. Many amino acids linked by peptide bonds will form a polypeptide. Furthermore, if we want to understand more about polypeptides, we say that these are long chains of amino acids which can be called as proteins. The only thing is all polypeptides are not yet ready to be called as proteins. Sometimes they need to undergo several processes of maturation to become functional. Now, how will these polypeptides form? Just like how we had studied how other polymers, other biological molecules form by polymerization, as in carbohydrates, amino acids will join by condensation reactions and they will form these polypeptides. Now, we have seen that amino acids have a carboxylic acid group and an amine group. So the C of the carboxylic acid group of one amino acid or the first amino acid will form a peptide bond with the N of the amino group of the next amino acid. We will soon be seeing the diagram. So remember we had studied about the peptide bonds using the beaded necklace. So these peptide bonds are nothing but covalent bonds, and since they are formed by condensation reaction, that means we will lose water during the process of formation of polypeptide. So amino acids will join one after the other in a linear fashion, and with each peptide bond that is being formed, water will be lost. One molecule of water will be lost. And here is the diagrammatic understanding of what we just discussed. That is, let's say we take the simplest amino acid, that is glycine. In glycine, the R group is nothing but just a H atom. So here the central carbon is bonded with an amine group and a carboxylic acid group. This is a second glycine. They align each other side by side so that the first glycine's carboxylic group faces the second glycine's amine group. Then the carboxylic group will lose its OH, the amine group will lose one of its H to release a water molecule. And now this carbon and this nitrogen will be having extra electron pairs to share with each other. So they will form a covalent bond where they will share their electrons, and this covalent bond is called a peptide bond, which is found between the carboxylic acid group's carbon and the amine group's nitrogen. So a single peptide bond joining two amino acids will lead to the formation of a dipeptide. So in a dipeptide, there is a single peptide bond. That's the beginning of how polypeptide forms. So a third amino acid can join over here. Similarly, a fourth. And as a new amino acid keeps getting joined, a new peptide bond will be formed. Here in this video, I now want you to pause and answer a few check for understanding questions. So my first question to you is, if you look at this structure in this reaction, how many amino acids are there in this structure that I am hovering my cursor on? Thank you for pausing. So I can see that there's a central carbon, amine group and carboxylic acid group here. So this is one amino acid. This is the next one. This is the third and this is the fourth. So I can see four amino acids, and if I'm asked how many peptide bonds are possibly going to form with these four amino acids, the first prompt response by mistake sometimes comes out as four peptide bonds, but you're wrong if you thought so. If there are four amino acids, three peptide bonds will be formed to join them. So look at the product over here. You can see that the red lines show the peptide bonds in the product. And with every peptide bond, one molecule of water will be released. So therefore, for this tetrapeptide, three water molecules will be released. Now I will ask the second question and you will again pause and think. My second question is, what if one of the amino acids, say this one, okay, what will happen if I flip it along its horizontal axis? So if I flip this amino acid in this manner as shown in the arrow, what will happen? I hope you can anticipate what problems will arise. So the problem that will arise if this amino acid is flipped horizontally is that this amino acid's amine group will face again the amine group of the next amino acid. The amine group and amine group cannot form a peptide bond. The peptide bonds in a polypeptide will always be formed between the carboxylic acid group and the amine group. Look, carboxylic acid group and amine group. If I flip the molecule, amine and amine group will repel each other and not form a peptide bond. So once again, let's quickly check what a peptide bond actually means. It actually means the bond between the C of the carboxylic acid group and the N of the amine group of two different adjacent or consecutive amino acids. And as you can see, as the peptide bond forms, this carbon loses an OH group and this nitrogen loses a hydrogen, and the OH and the H combine and form a water molecule. Next fact about peptide bond is that they are covalent bonds, and every time you study biomolecules, try to build a relationship with your past knowledge. So previously we studied about lipids and carbohydrates, and there also we had studied about two covalent bonds, that the ester linkage and the glycosidic bond. The third covalent bond that we are studying in this unit is the peptide bond. A polypeptide is nothing but a polymer, and it is made up of more than 3 amino acids. To be more specific, if there are more than 10 amino acids, we call it a polypeptide. If it is between 3 to 10, it can also be called as an oligopeptide. Polypeptide range in length from few to more than 1000 monomers. So at least there should be 10 amino acids or more than 3 amino acids to call the molecule a polypeptide. But there are polypeptides who have thousands of amino acids linked together in a chain. Each polypeptide can have a unique linear sequence. So we know there are 20 different amino acids. These 20 different amino acids can align themselves in any order, any number of times they can repeat. So multiple thousands of polypeptides can be formed by different combinations of these 20 amino acid types. But every time a polypeptide will be formed, it will start with either an amino group or carboxyl group and end with an amino group or carboxyl. So let's understand what this C terminus and N terminus thing is. Let's say a peptide bond is forming. Look over here. So there are two animations happening side by side. The first animation, let's wait for it, is condensation. So here you can see that a water molecule comes and is getting released and a peptide bond is being formed. In hydrolysis, on the other hand, the peptide bond breaks. That is, the OH will again bond with the COH and the H here binds with the NH group and forms a amine group. So both condensation and hydrolysis can happen to form or break down the peptide bonds. So you can pause and take a look at what's happening in the condensation reaction and what is happening in the hydrolysis reaction. In condensation, a polypeptide will be formed. In hydrolysis, the polypeptide will break into different peptides or different amino acids. Here is a still image of how condensation happens. Condensation is also referred to as dehydration synthesis, means water is getting removed, so it's a dehydration reaction, and because of the water removal, C can bond with N and form a peptide group. Now look at what we were talking about, the amino terminus and the carboxy terminus. Every time a linear chain of polypeptide is formed, both ends will have the two functional groups. Either there will be an amino group here and a carboxyl terminus here, or sometimes it will be the other way around in a 3D space. So the amino terminal has the amine group, so it's also sometimes called as the N terminus. Since the carboxy terminus has the carboxylic acid group, it is also sometimes referred to as the C terminus. No matter how long this chain keeps growing, there will always be one N terminus and a C terminus here. Now, I hope you have your notebooks and pen with you. So I would like you to pause the video, and just like you saw the condensation reaction diagrammatically, you will similarly take notes or sketch notes on how the hydrolysis reaction of peptide bonds look like. So you may choose to take a dipeptide and show what happens to that dipeptide when it hydrolyzes. Thank you for pausing and sketching your note. With this, we finally recap that polypeptides are nothing but a chain of amino acids linked together by peptide bonds. Sometimes we can call a polypeptide chain as a protein. A protein actually consists of sometimes just a single polypeptide chain, or there are complex proteins which are made up of more than one polypeptide chains. So if you take many polypeptide chains together and if they are attracting each other, they will form a sort of a interaction and fold themselves in a manner in three-dimension shape to either become a rope-like structure or a ball-like structure. So we will study about these structure in the next task. So this is the key focus, that polypeptides form the main structure of the proteins, whereas the polypeptide themselves are made up of amino acids. Proteins can be formed in 4 levels of structures, that is primary, secondary, tertiary and quaternary. We are going to study about these structures in the next task. With that, we come to the end of this lesson where we learned about what polypeptides are, how they can be formed, how they can be task. We will be studying more about the primary, secondary, tertiary and quaternary structure of proteins. Thank you.